Hello all, welcome to the second session of the day. And so today our speaker is from Ross Lifesciences.in. I have posted the website of the companies of experience in agrochemical and public health field. On urban mosquito control. So the mosquito control is one of important portfolio of Dr. Kedar, and his company they also do toxicology and food related testing services, and they also it's a very dynamic company based in Pune, but they have their office in outside India also. They have a part of their office in located in United States. So today's the speaker got some auditing in the morning. So he called me and said, there is some urgent audit came in my office today. So, typically what people at this stage will do is can you reschedule my talk? Can you do something like that? But what he had done is he recorded his talk and then gave it to us. I don't want you to change the schedule or something like that. Please go ahead with my talk, and I will come back later for the QA question answer session and discussion session. So with this, I will share my screen. I will.
Good afternoon. At the outset, I would like to thank the management of ARI and PKC for giving me this opportunity to briefly talk about the topic. As an example, I would be highlighting activities of Ross Life Science that would give you some flavor on varied domains that exist in an enterprise like a CRO. After the
Sheetal or Shilpa, if somebody just check if something misses or the volume is low or something, please let me know.
It's fine. You can just do that, right?
Okay. Thank you.
Presentation. You would appreciate various opportunities that are available for biotechnologists or life science students to make their career or start a new enterprise of their own. Ask yourself a question. What is my passion? If you dream of making a difference in the world by developing a life-saving vaccine or medication, or maintaining the quality of foods to prevent widespread illnesses, or pushing society into the future with breakthroughs in technology, a career in life sciences would offer you all these opportunities. Life sciences are usually at the forefront of scientific breakthroughs in medicine, but they have also held crucial roles in other fields, such as agriculture and food protection, for several years now. I would like to give you an example of various activities pursued by Ross Labs in various domains of life sciences, which would give you enough idea on career opportunities that exist in the field of life sciences. If you can take some inspiration from this presentation today, then I would say that the objective is served. Ross Life Science was started in Pune in the year 2008 and is named after Nobel laureate Dr. Ronald Ross, who showed how malaria is transmitted from mosquitoes to man. Ross also stands for research-oriented systemic studies. Vision for Ross Life Science is to be a leading contract research and manufacturing services organization for creating innovative customer-focused solutions, offering cutting-edge technology in the field of integrated pest management, home care, and personal care. As a CRO, there are three main pillars of activities that we are engaged in. One is registration data generation of crop protection, household insecticide, and public health products, wherein Central Insecticide Board and Registration Committee is the apex body for granting registration for pesticides in India. The second activity deals with testing and evaluation of products from various categories, as is depicted in the slide here. This activity deals with assessing performance of the products and also for substantiating marketing claims. For example, several times faster knockdown of insects as compared to a particular variety, or 99.9% germ kill claim, etc. The third pillar of activity involves formulation development, which includes emulsifiable concentrates, which are most common and simplest type of formulations, apart from suspensible concentrates, encapsulated and nano formulations. Packaging development plays a critical role in rendering the product withstand rigors of transportation and also making the product stand out in the shelf among clutter of other products. Novel delivery mechanisms for focused and targeted application on insect pests is critical for optimum performance of the product. This involves aerosol cans with attached straw for spraying in cracks and crevices or cockroach control. As you can see from this slide, contract research and testing is comprised of multidisciplinary activities wherein the products undergo exhaustive and comprehensive evaluation in terms of chemical analysis, packaging, biological performance of the product against target insect pests or microbes in question, toxicological assessment on the effect of pesticide products on beneficial and non-target organisms. This requires expert and experienced manpower in each of these disciplines and speaks about job opportunities available in this sector. The slide showcases other activities that we conduct, which includes product development that meets clients' brief, allied testing services, other than crop protection, household insecticide, and public health, manufacturing services, wherein we source raw material and packaging material and convert them into finished products for supplying them to our clients in timely manner without compromising quality or regulatory norms. We also have ROS Innovation Cell for development of in-house innovative and patentable products with cutting-edge technology. Certifications, are important and crucial part of any CRO to conduct its operations as it gives credibility to the data that is generated. Obtaining certifications require excellent infrastructure, systems and processes for it to qualify for that kind of certification. ROS is now a GLP certified lab, which means that the data generated is accepted by regulatory authorities across all OECD countries. ROS is also NABL accredited, DSI recognized, and also approved by Registration Authority in India, which is Central Insecticide Board and Registration Committee. Typical product development passes through various stage gates, from ideation to commercialization, and involves filtering of ideas through ideation session with the client, working on selected ideas to develop the product from bench scale to commercial scale. I will now just rush through slides that give flavor of activity in chemistry and other fields. In analytical chemistry, we conduct 5-batch analysis for ascertaining the purity of insecticide and other impurities and conduct characterization of technical or formulated product for various physicochemical characteristics. We also conduct air concentration study to estimate how much of active ingredient is delivered by various devices like coil, liquid vaporizer, aerosols, and to determine degradation of insecticide at various intervals of time, which enables one to predict longer lingering effect of pesticide in ambient environment. We perform residue analysis of pesticides in plant, soil, and water. The objective of residual analysis is to estimate presence of pesticide on plants, in soil, and in water, and to study the degradation pattern at various intervals of time. The residues on plants allow one to determine whether the pesticide residue levels are within tolerable range and whether the crop is fit for consumption or not. Once a pesticide has been released into the environment, it can be broken down by various means. Exposure to sunlight, which is called photolysis, exposure to water, which is called hydrolysis, exposure to other chemicals via oxidation and reduction mechanisms, microbial activities through bacteria, fungi, and other microorganisms, or by plants or animals through their metabolism. We do experiments to determine how long pesticides last in various environments which are called EFET studies. We apply pesticides to soils, leaves, or other surfaces and measure the time it takes for half of the pesticide to break down. This measure is called half-life. We also conduct stability studies at various temperature and humidity conditions as per ICH and/or WHO guidelines to determine whether the product and package is compatible with each other and whether the formulation is stable over a stated period of shelf life claim. Most of the products declare 2 years of shelf life claim on the label. We also perform real-life stability studies wherein the product is stored at different geographical regions at ambient temperature and check for physicochemical properties at different time intervals to see whether the product conforms to the specification or not. Packaging trials include various tests like drop test, vibration test, inclined impact test to check whether the packaged product can withstand rigors of transportation and handling at various points of loading and unloading and stacking at the warehouse. We have excellent entomology facility wherein we employ agriculture and public health entomologists to conduct laboratory and field work. We have very unique entomology facility wherein we rear various insects like mosquitoes, cockroaches, houseflies, bedbugs, and stored grill pests. The species is confirmed by DNA barcoding, and mosquitoes are reared on artificial blood feeding apparatus. We have sophisticated chambers of various sizes to conduct performance of products against target insect pests. We conduct both laboratory and field trials for evaluating efficacy of products against pests of public health importance. Likewise, we conduct field trials for crop protection chemicals in various geographical regions of India for various categories of formulation, whether it's insecticide, herbicide, fungicide, or plant growth regulators. The studies are conducted as per the client's need and reports submitted with statistical analysis. Ecotoxicity studies are conducted on various beneficial organisms to determine the impact of chemicals or pesticides on them. The studies are conducted on fish, avian, alga, honeybees, daphnia, and earthworm as per OECD guidelines. There is a lot of interest in honeybees as they are adversely impacted by use of neonicotinoids, which are a class of insecticides used in agriculture on vast scale. We also conduct field trials to see the impact on various pesticides on reproductive potential in earthworms and honeybees. Genotoxicity is the term that describes property of chemicals or pesticides pertaining to their ability to cause damage to genetic material. Genotoxicity can lead to direct or indirect effects on DNA, including mutation, induction, mistime event activation, and direct DNA damage, which leads to mutations. The battery of assays conducted by us include EMS test, wherein the endpoint is gene mutation in bacteria, and in vitro with cytogenic evaluation of chromosomal damage with mammalian cells, or an in vitro mouse lymphoma TK assay, and micronucleus test, which detects nucleus aberrations, that is formation of small micronucleus in addition to the main nucleus. We have recently entered into varied testing services in the realm of food analytics, pharma, and other allied testing services, which is more to do with analysis and characterization of physicochemical properties and some testing related to microbiology in case of food. The other area which is unique to our kind of CRO is we develop innovative patented product and supply it to the client in timely manner, meeting all regulatory and quality norms. We source raw material and packaging material at cost-effective rates and convert it into finished product at the third-party manufacturing unit for supplying it to client. Finally, there are innumerable opportunities for life science students in the disciplines that are highlighted in the slide. I hope the presentation would have given insights into opportunities that are available today in CRO business. I will leave you with two examples of new technology that we are working in the field of molecular entomology, which has huge implications in vector control, and another one which involves a cell line assay to find out whether the compound or molecule is indeed endocrine disruptor or not. We have entered into strategic alliance with Israel company on sterile insect technology for control of mosquitoes that spread deadly diseases like dengue and chikungunya. The technology utilizes gene silencing by use of double-stranded RNA of gene that encodes polypyrimidine tract binding protein responsible for male fertility. Treatment with double-stranded RNA at larval stages renders adult male sterile. Massive and sustained release of sterile male mosquitoes mate with wild females to yield less viable offspring, resulting in reduced mosquito population. This is yet another research project that we are jointly working with Israel company, wherein mosquitoes resistant to synthetic pyrethroids are rendered susceptible by silencing the gene for voltage-gated sodium channels of the nerve membrane. This has huge implications for vector control, as the need for discovering new molecules is obviated, which is not only expensive but also time-consuming. We have recently initiated cell line assay involving HeLa 9903 cell line for endocrine disruptors, which are chemicals or pesticides that interfere with endocrine systems and can cause birth defects and other developmental disorders. In this assay, endocrine disruptor chemicals bind to estrogen or androgen receptor to form a complex. This complex then binds to DNA responsive element, which induces the expression of reporter gene, that is luciferase. The luciferin thus formed is measured by fluorescent microscope to confirm whether the compound is endocrine disruptor or not. To summarize, the global CRO market is $53 billion presently and is growing at a CAGR of 11%, whereas the Indian CRO market is around $900 million and presents immense opportunities in the fields mentioned in this slide, apart from clinical research. Finally, to quote Jack Welch about winning in business is great because when companies win, people thrive and grow. I would like to end this presentation with a short film which briefly describes the activities of Rouse Lifesciences.
Rouse Lifesciences Private Limited is a contract research organization based in Pune and has been operating since 2008. It is engaged in product development and analytical services, including registration, data generation, especially in the field of integrated pest management, which involves crop protection, public health, household insecticides, and veterinary ectoparasiticides and home care. Notably, Rouse Lifesciences is a OECD GLP approved laboratory, which means that the data generated by us under this protocol will be valid in 36 member countries, including the US, several countries in Europe and elsewhere, which are signatory to this international organization. Rouse Lifesciences has experts and expertise in the field of organic chemistry, entomology, ecotoxicology, and microbiology. The laboratory infrastructure is state-of-the-art for conducting prototype development, product testing, and analytical studies. A technical advisory panel comprising of professors from academia are accessible to help guide with the latest technical developments in the field. The entomology division of Rouse Lifesciences has expertise and competency for conducting assessment of various insecticide products against target pests, both in lab as well as field. We have in-house facility for rearing insect colonies of household and public health importance, along with colonies of stored grain pests. The range of insect colonies include susceptible and resistant species of mosquitoes, houseflies, and roaches, as well bedbugs. The entomology facility is equipped with chambers of various sizes for evaluating efficacy of insecticide products. It consists of peat Grady chambers and 30 cubic meter large chambers with air exchange facility, direct spray knockdown chamber, and also residual efficacy room for storing various panels. We also routinely conduct release rate studies of various product forms in our evaporation chambers. Rouse has successfully executed dozens of field trials with special mosquito repellents using household formats like coils, mats, aerosols, liquid electrics, and burn paper, as well as personal mosquito repellents using sprays, lotions, etc. It also has worked with pest control operators, MNCs, and other organizations engaged in vector control for testing products like larvicides, thermal fogging concentrates, and IRS, etc. Chemistry facility of Ross Life Science includes various activities like new product development, analytical chemistry, stability studies, including container content compatibility studies, residue and persistency studies, and packaging studies. We have conducted several studies relating to 5-batch analysis of active ingredient and its formulation as per the international guidelines using instruments such as GC-MS, HPLC-MS, MS, FTIR, etc. The facility also has well-equipped storage stability chambers to conduct shelf life studies as per the CPAC, or Collaborative International Pesticides Analytical Council, as well as WHO and local regulatory guidelines. The microbiology department of Ross Life Science is involved in activities related to evaluation of biopesticides, disinfectants, and air sanitizers for assessing performance of products. Welcome to Ross. Welcome to life.
Thank you all. We'll try to get the Dr. Kedar to talk to us sometime, when he finds time. So we'll get him for the interaction session. So with this, we are done with this week. So our next session is on 18th. That's on Monday morning. So because of Dasara tomorrow and then Saturday, Sunday, we don't have any session. So we will resume back on Monday morning, 10 a.m. So is any of the participants, okay. Somebody asked, sir, do you provide the insect larva for research to other institutions? Maybe I can get in touch with Dr. Kedar, or like we can get him here sometime, maybe in the next week, we'll try to schedule some meeting with him. So that time we can ask these questions. So we will meet on Monday morning, 10:30. So there is no sessions on Friday, Saturday, Sunday. So we'll meet on next Monday. Is anybody have anything to share or something like that? Any of the participants want to share your concern or remarks or some feedback, whatever happened till now? Either you can unmute yourself and talk, or you can put it in the chat box. We'll try to address those. Okay. Somebody says, excellent session. Thanks. So we just, started the next week is going to be a little bit more intensive. And another one on 20th. So 18th and 19th we have session. On 20th, we are going to have only discussion among ourselves. So again, we will be making breakout rooms on that day. So please, be prepared to come and talk on that day, just to discuss, who your fellow participants. So if you have the course scheduled with you, 18th we have one session, 19th we have two sessions, and 20th we don't have any sessions planned on 20th. So it is going to be just a recap of whatever happened in the last few days. And if possible, we'll plan for some quiz program or something like that. And then it's going to be interaction among the participants, with some of us will be around there. So with this, we'll close the session today. And those who are part of that group, WhatsApp group created by the participants, please post there also. Our next session will be on Monday, 10:30. The same link will work. Thank you.